Hi everyone, welcome to the session of INI difficult questions discussed just before your exams. So let's move to the first question. A neonate was normal at birth but developed liver disease and muscle weakness at 4 months. She had severe hypoglycemia. So it is saying that it can be a case of liver glycogen storage disorder, particularly in the morning. So definitely it is a fasting hypoglycemia. So it is a liver glycogen storage disorder most probably. On examination, there is hepatomegaly. So again, it is saying that it can be a liver GSD only. Lab test done after fasting shows ketoacidosis. Then pH is reduced. So it's a part of ketoacidosis only. Increased ALT and increased AST levels are there. That is liver is affected. On examination, IV Glucagon challenge after carbohydrate meal, a normal rise in blood glucose level is seen, whereas and the liver biopsy showed increased glycogen content. So there are two diagnoses which come to your mind. Is one is von Gehr's disease. So if you see, there are many things which is favorable for von Gehr's disease. For example, in this case, ketoacidosis is there. So there is metabolic acidosis, then hepatomegaly, then as usual in the case of fasting hypoglycemia is there. Now, when you compare it with this Cory's disease also, there are some features of Cory's disease as well. For example, hepatomegaly is there in Cory's disease as well. Then when you see that, you can see liver biopsy, it is showing glycogen content is increased. So there is a trap there. That is, it is not mentioned whether it is normal glycogen or abnormal glycogen. So if it is abnormal glycogen, it is more towards Cory's disease. Normal glycogen, then it is more towards von Gehr's disease. So if there are features which are inconclusive of we are not sure what is the disorder, then most probably what we'll do is a test called as IV Glucagon challenge. So we will see what is the result of an IV Glucagon challenge. Always remember, this IV Glucagon challenge is done in two situations. One is after an overnight fast and the other one is after meals. So we'll think about an after meals. So in situation where it is an after meals, we know that what is the hormone in the body? It should be insulin. So on the top of that, what we are giving is glucagon. So just think, like if there is glucagon, we know that it is going to increase glycogenolysis as well as gluconeogenesis. So already there is insulin and the body is in a well-fed state or after meals. So we know that the blood glucose level will be there. So on top of that, what we are giving is a glucagon, a bolus of glucagon. So what is the action of glucagon? It is going to increase the gluconeogenesis and is going to increase glycogenolysis. So if it is going to increase the glycogenolysis, what will happen to the blood glucose level? It should rise, right? Now we are going to differentiate between von Gehr's and Cory's disease. So in Cory's disease, what happens? Where is the defect? In Cory's disease, the defect is in a debranching enzyme. So if there is a defect in debranching enzyme and if you are giving a glucagon challenge, we know that glycogen phosphorylase activity is increased. So because of that, glucose 1-phosphate is released and we know glucose 6-phosphate can be formed and glucose can be formed. Only thing is that all the glucose cannot be released from glycogen because the debranching enzyme is not there. But still there is possibility that some glucose can be released from glycogen and this can be converted to glucose, which means that if this child is having Cory's disease, then you can see there is a rise in blood glucose. So if there is a rise in blood glucose with IV Glucagon challenge after meals, it says that it is Cory's disease. Now what about von Gehr's disease? Where is the defect? The defect is in glucose 6-phosphatase. So already blood glucose is there and we are giving a challenge of glucagon and it's trying to increase glycogenolysis. So definitely this is going to get increased. The activity is increased. Glucose 1-phosphate is produced. But what is the trap there? This glucose 6-phosphate cannot be converted to glucose because the final enzyme is not there. So will there be rise in blood glucose? No, there won't be rise in blood glucose. So there is no rise in blood glucose. So I should write here, there is no rise in blood glucose, then it is a case of von Gehr's disease. Now you see this question, there are many features of Curies disease as well as von Gehr's disease, and we are not sure what is the diagnosis. Then we see IV Glucagon challenge, can see that after overnight fast, there is no rise in blood glucose, but what you can see after a meal, normal rise in blood glucose level is seen. So which means that this is a case of Curies disease and the enzyme defect is debranching enzyme. Now I didn't tell you anything about the next thing which is given here after an overnight fast. What is, so after an overnight fast, remember that overnight fast means the body is already in a glucagon stage. That is, glucagon level is high in an overnight fast. So on top of that, we are giving a bolus of glucagon. It's not going to increase anything because already we are in a glucagon stage, and even if we give an IV glucagon challenge, then nothing much is happening there, which means that there is no rise in blood glucose, and we cannot conclude anything from the IV glucagon challenge in overnight fast, so that we can't differentiate between Curies and von Gehr's disease by doing an IV glucagon challenge in when the body is in an overnight fast. So always think only this is significant here after meal. So rise in blood glucose means it is Curies disease. No rise in blood glucose means it is von Gehr's disease. So that is how the answer came to be debranching enzyme. So we'll move to the next question. It is the question on titration curve. As you all know, I have already discussed about titration curve in the part 1 of difficult questions of INI. In this question, I'm just confining to what is asked in this exam. That is, the question asked is what is Zwitter ionic pH? Zwitter ionic pH is otherwise isoelectric pH. That is what we write as PI. How to calculate PI? PI is equal to pK1, which is the ionization constant of the first ionizable group, plus the ionization constant of the second ionizable group divided by 2. So here when we add and divide it by 2, we'll get the answer as 6.1. So that is how the answer came to be 6. Now we'll go to the next question. Which method are used to identify different mutated strains of same species of Mycobacterium tuberculosis? RFLP, chromosome mutation detection, Spoligotying, and multiple interspersed repetitive unit typing, or MIRU VNTR. So this is about strain typing, that is genotyping of strains in bacteria. We know that the strains are happening because of different mutations that is happening. So there are different strains of the same bacteria. We know that. So how to identify the strain? Each strain of this bacteria is differing in certain mutation or certain there are certain genomic variation between these different strains. How to find it out? So for that, what the technique that we use is generally what we call it as the chromosome mutation analysis. So it is a term which we use to include all the different techniques that we use for what is called as a strain typing of bacteria. So that is called chromosome mutation analysis. So how to identify it, or what are the different techniques here there? So the first one under that is restriction fragment length polymorphism. You all know what is restriction fragment length polymorphism. I have uploaded in a video on RFLP also. So RFLP is a older technique where we are dividing, or we are finding out what is the different mutations that is happening in two different strains so that we can identify a strain. This is an older technique. So this is replaced by newer technique like Spoligotying. What is Spoligotying? So for that, to understand that, we should know something about the genetics of a bacteria. Particularly in this case, it's a Mycobacterium tuberculosis. So we should understand that in a bacteria, there are certain regions in this DNA which are called as the direct repeats. So apart from that, there is a spacer region between these direct repeat. And these spacers are unique for each strain. that is the base of doing something called as Spoligotyping. So if we are identifying these unique spaces in a strain of bacteria or different strains of bacteria, that is called as Spoligotyping. Then the next one is multiple interspersed repetitive unit, which are what is called as, you all know what is variable VNTR, that is variable number tandem repeat, that we have learned in humans also. The same thing is that one strain of a bacteria is differing from other strain of bacteria in a certain normal genomic variations. They are called as the variable number tandem repeats, which means certain sequences are repeated at different numbers in different strains. So that is called as VNTR. Now if we are directly typing these VNTR, then it is called as MIRU typing, otherwise multiple interspersed repetitive, that is the VNTR, variable number tandem repeats. These are repetitive units are being analyzed or typed, which is a direct typing, and that is called MIRU VNTR or MIRU typing. Then what are the other techniques? Newer techniques are there, because if we have lots of genome like nucleotide sequences are with us, or different samples of the strains are there, then we have to use some next generation, like newer techniques like next generation sequencing. So under next generation sequencing, what we can do is we can find out the whole genome of, or we can analyze the whole genome of a bacteria or a bacterial strain, that is called whole genome sequencing. Or we can sequence only the coding regions, that is called whole exome sequencing. So in this case, it is the whole genome sequencing, and whole exome sequencing is coming under next generation sequencing. So all these are different techniques to type the different strains of the bacteria. So what is the answer here? Restriction, fragment length, polymorphism, yes. Chromosome mutation detection, yes. That is a term that we use to include all these techniques. Spoligotyping is there and MIRU is there. So all these are answers. So the answer here is 1, 2, 3 and 4. So we'll go to the next question, which was asked in the last INI. This question is about what is the technique. That's an image is given and asked what is this technique. So first and foremost, you see the option to get some clues for this question. So it is regarding something with the antigen antibody reaction. So it is given that two options are of ELISA and the two options are of something else, which are called as surface plasmon resonance. So if we understand that it is not ELISA, then we have to differentiate between two, or we have to find out the answer from two options only. So first we should know what is an ELISA. ELISA definitely understand that it is like something which the antigen antibody interaction has taken place, and then some labels are added, and this label is giving us some color or a reaction, something like that. It is given by this label, and that is what we are detecting, and we are quantifying the concentration of the antigen or antibody in the sample. This is called as ELISA. So it is like an endpoint method where we are detecting the concentration of the antigen after antigen antibody reaction has taken place, just like a conventional PCR. But what about the next thing that is given as a surface plasmon resonance? It is like a real-time PCR. You know what is real-time PCR? Detection and amplification is happening simultaneously. So this is something like that where we are detecting the antigen antibody interaction as and when it is occurring. This is called as surface plasmon resonance. So if you see this, if you see this, what you can see that, so initially there is no antigen antibody interaction, so there is no surface plasmon resonance, and later on what you can see is that gradually the surface plasmon resonance curve is rising. Then it is in the later stage, what you can see that once all the antigen antibody complexes are formed, it is peaking and then it is coming down. So this is a real-time method where we are detecting the antigen antibody interaction, and thereby we are detecting the affinity of the antigens towards the antibodies real time. So basic technique that we want for the surface plasmon resonance is that we need a light source and there is a detector here. Now when this light source is falling on a glass prism like this, and you can see that this antigen antibody interaction is happening here. So once when the antigen antibody interaction is happening, so initially there is, this light source is falling on this glass prism. Now at a point where antigen antibody interaction is maximum, what you can see is that certain waves are created. They are called as surface plasmon resonance waves are there, and this waves can be detected, and you can see that this light which is falling on this glass prism is deflected and we can detect it. So maximum detection is possible when there is maximum interaction between the antigen and antibody. So how it is detected? Because of certain resonance waves that is being created by the interaction of this antigen and antibody. So this is the basic principle of what is called as surface plasmon resonance. So coming to the answer, definitely it is not an ELISA because it is not an endpoint method. It's a real-time method. And this is surface plasmon resonance for antibody affinity. It is not antibody concentration. We are not measuring the concentration of the antibody here. We are measuring the affinity of the antibody towards the antigen. So here the answer is C. So next we'll see another difficult question asked. Which of the following mutation result in reversal to wild type of phenotype so that mutant gene is suppressed? So we should understand that what is the meaning of this wild type of genotype. Wild type of genotype means the normal genotype. So if there is a allele there are two types of alleles, or a gene there are two types. That is the wild gene or the mutant gene. So wild gene, which is the normal, mutant means there is some base sequence variation or deletion or insertion. Then it is called as mutation. So here the mutation is suppressed because of something else. So that is, what is that is being asked here. So before coming to the answer, let me discuss about something called as tRNA suppressor mutation. tRNA suppressor mutation means, just see this picture. So here we know that UAC, this is a codon for tyrosine. Now this UAC is binding to a tRNA which carry tyrosine. And in the anticodon arm, you can see that there is a complementary sequence, that is GUA, which can bind to this UAC. This is normal process. But a mutation happened to this DNA in such a way that, and correspondingly in the mRNA, in such a way that this UAC has been changed to UAG. We know that UAG is a stop codon. So this is an example of a nonsense mutation. So what actually should happen is that this translation process should stop because a stop codon has been created in between. But it will not stop. How? So for that you can see that we know that UAC is supposed to bind to the GUC. Some mutation or base substitution happened in the anticodon in such a way that this G has been changed to C. So this tRNA which carry tyrosine in its acceptor arm, will it bind to this amber codon or the stop codon? Yes, it will bind, right? So even though a mutation has happened in the DNA and correspondingly in the mRNA, the tRNA is acting smart in such a way that a base substitution happened in the tRNA so that even if it's a stop codon, it is becoming a coding codon because there is a base substitution in the anticodon arm of the tRNA. This is called as tRNA suppressor mutation. So what will happen? That is, the mutant gene is suppressed so that it is reversal to the wild type or normal is happening because of this tRNA suppressor mutation. So now we'll see each of the options. Deletion of mutated gene, no. Frame shift of mutated gene, no. Addition of another normal gene, no. Mutation in the tRNA, yes. That is the answer here. So this is tRNA suppressor mutation. So going to the next question, what is the effect of RNAi on gene expression? So the answer here is knockdown. I have already uploaded a video on microRNA that is discussed in just 3 minutes. If you're not, if you haven't watched it, you can watch that also. So here I will tell you about this microRNA, the most important points that you should learn about microRNA. Number one, it is having an endogenous origin from a prime microRNA gene. Number two, this is undergoing extensive post-transcriptional modification in such a way that initially you can see that it is a double-stranded, there is a poly A tail here, you can see that there is a loop there. What happens? There are some endonucleases are acting. So inside the nucleus itself, an endonuclease called as DROSHA is acting, and it is cutting that poly A tail. So then after that it is exported out. Once it is out, it is again acted upon by another endonuclease called as dicer, so that loop is also cut. So it becomes a double-stranded microRNA. So this is a microRNA which is double-stranded. Now this is loaded to a protein complex which is called as RNA induced silencing complex, that is RISC. This is made of proteins called as argonoate proteins. So it is loaded. There are two strands in this RISC. What happens later is that one strand, you can see that one strand is cleared. Now it is having only a single strand. Now this is called as a completely mature microRNA, where it is a single stranded non-coding RNA which is 21 to 22 nucleotide length, and it is a single stranded RNA. This is a microRNA. Right. Now what you can see here is what is the action of a microRNA. Inside this RNA induced silencing complex, this microRNA can bind to an mRNA, where it is binding to the 3'. That means at the 3' end of the microRNA, there is a region called as UTR. UTR means untranslated region. So there is at the tip of that 3' end, you can see that there is a region which is not coded to the protein. It is an untranslated region. So there is a sequence in this untranslated region. This is again another question. To this 3' untranslated region, this microRNA is binding. Now what is the action of this? There are two possibilities. You can see that if this binding is in such a way that it is completely or it is a perfectly binding, then it will cleave the mRNA. mRNA is cut and it is destroyed. Now, if the pairing is an imperfect, then it cannot cleave the RNA or the mRNA, but it will suppress the translation of this mRNA. So it is causing translation arrest. So whatever is happening here, this is a post-transcriptional modification, and this process is called as RNAi, otherwise RNA interference. And this is otherwise called gene knockdown. Okay, so that is about the microRNA.